Hello, I'm David Hodgson, and the paper that I've decided to look at is autophagy is a protective mechanism in normal cartilage, and its aging-related loss is linked with cell death and osteoarthritis. So I'll just start by telling you a little bit about osteoarthritis. Osteoarthritis, or OA, is a disease that is characterized by degeneration of cartilage and its underlying bone within a joint, as well as bony overgrowth. It's majorly affected by age. Most people develop it if they live to a very old age, and it is in fact the most common age-related joint pathology. Unfortunately, the mechanisms leading to cartilage degeneration still haven't totally been determined, but it is known that articular cartilage has a low turnover of extracellular matrix cells. That makes them very susceptible to changes associated with age, and mechanisms that allow for cellular damage to be repaired is very important for their survival, and that's why they think it's more effective with age, because obviously there's more damage. Knowing this, the paper they decided to look at macroautophagy, as it is important for the turnover of intracellular organelles and molecules that protect the cell during stress responses. Recent studies have also shown it slows down as we age, so they think that it could therefore be very important in osteoarthritis. So in order to test it, they had to have something to look at. So they picked three important proteins for macroautophagy. The first one is ULK1, which is a key intermediate in the transduction of pro-autophagic signals to autophagosome formation. The second one they chose to look at is Becklin-1, which is a regulator of autophagy. It is a key intermediate. It forms a complex with two other proteins, type 3 P1I3 kinase and VPS34, that allow the nucleation of autophagic vesicles, which just means it allows their different vesicles to travel into the nucleus. Finally, LC3 is the other protein they looked at, and it's present in two forms: LC3-1 in the cytoplasm and LC3-2 bound to the autophagosome membrane during autophagy. LC3-1 is converted to LC3-2 through lipidation by ubiquitin-like systems, resulting in the association of LC3-2 with autophagy vesicles. They used many techniques during this paper, but the ones that I felt were the most important were immunohistochemistry and Western blotting. They were very important throughout, as they allowed them to identify the proteins and also show where they were expressed. But not only that, they managed to give them some measure of quantification so they could compare them all the way through on their different studies. You don't really have time to go through exactly the methods they used, but they pretty much followed a normal standard procedure for immunohistochemistry and Western blotting. They also used a lot of other techniques, such as culturing, collecting tissues, statistical softwares, and all sorts of quantification techniques. So here's the first results they found. The top part A is a Western blot that shows just normal tissue, mild osteoarthritis tissue, and osteoarthritis tissue. It's taken from human samples that have been collected after donation from a cartilage removal surgery. Just GAPDH is something I haven't mentioned before. It's just there as a control to show that the levels are pretty much similar through all, and it allows them to normalize their different levels before they quantify them. So as you can see, by B, C, and D are just different graphs to try and show better to try and show the quantifications better. As you can see, for most of them, in mild OA, it doesn't really go down too much, except for the IC3 levels. But in osteoarthritis, all three are statistically importantly lower. They are a lot lower, which is important. They then they moved on to do a mouse test, because it was easy with the different time frames, as mouse don't live as long as humans. And they just wanted to look at how the levels of the proteins decreased over time. So they took samples from 3 different aged mice, when they were 2 months old, when they were 9 months old, when they were 12 months old. They only did it for Bacillin-1 and LC3, but as you can see, both of them reduced significantly in both 9 months and 12 months. Sorry, pardon me. So move on to the next one. Just to make sure that that the proteins were lower, due to the osteoarthritis and not due to just the proteins were lower in osteoarthritis. And this wasn't just because people who have osteoarthritis are older and therefore the proteins have gone down anyway. They performed sham surgeries on mice to induce OA when they were still reasonably young. I think it was around 3 months. I'm not sure. And then they measured the levels of bacillin-1 and ULK1 8 weeks after surgery. And then, as you can see, both of them are significantly lower, which shows that the reduced levels in the osteoarthritis isn't just because the patients are older than non-diseased. No, sorry, that makes sense. Just to show that the patient's levels of these proteins aren't lower just because they're old. They are lower because of the osteoarthritis as well. The final result I want to show you is they wanted to compare the relationship between autophagy and apoptotic cell death. And to do this, they performed immunohistochemistry on mice again, and they looked for a marker, ADP ribose polymerase, or PARP, as I'm going to call it from now on. They were shown from the two preceding experiments, 2 months, 9 months, and 12 months were just the normal age in mice samples, and DNA were taken from the sham surgery samples, performed in the same way as they were for the other ones. And you can clearly see there are similarities between C and E, that so more cell death means loss of autophagy, which may contribute to cell death. So just go through what they decided, what the study shows. Firstly, they show that autophagy may be important in normal cartilage, whether that be a protective or homeostatic mechanism. They then also show that the expression of 3 autophagy markers was significantly decreased in osteoarthritis tissue, and provided evidence that this wasn't just a result of the aging population. Their PARP studies also showed that this reduced in these proteins, that the reduction in these proteins led to increased apoptosis. They, however, feel that the further analysis is required to determine whether a direct causal relationship exists between the two processes in cartilage. The only caveat I would have with the paper, the question the results is, in both the first study in the Asian mice and the second study in the sham surgery, they only look at 2 proteins. In this one, they look at bacillin-1 and UKL1, but they miss out LC3, whereas in the second sham surgery one, in the aging tissue one, they have LC3 and bacillin-1, but they instead miss out ULK1. So although these probably will follow the same sort of system as the other ones, you cannot just say that without looking at it first. So I would say that they need to do them experiments really before they can totally justify their claims. But except from that, everything in the paper was really good. So if you have any questions, please just comment. Thank you.